Okay, hello everybody. So we are very happy to welcome you today for this afternoon symposia on the fetal foundation of neurocognition. And so we will have the great pleasure to welcome Petra Hüppi now, who coming from University of Geneva, and who will talk about preterm brain between nature and nurture.
Thank you very much, Anu. I'm very happy to talk to you today. I'm a neonatologist, so you will also get a little bit of a feel of what is actually a preterm baby, not just the brain. And I put together sort of some pressing questions that we have and some new results that we recently could show where we are interested in modulating brain development. So child development is really something that starts in preconception. It goes through the fetal and prenatal period, and then it also gets modified in early childhood, middle childhood, adolescence, and adulthood. During this time, biology of health is characterized by many physiological adaptations, but also disruptions. And these can be cumulative over time, and they are embedded usually in sensitive periods. So the core concepts of development for the human child is that it is the fundament of mental health later on in adulthood, that the brains are built over time, and that there are interactive influences of genes and experience that shape the developing brain. And therefore, it's important to think about biological as well as contextual risk factor or factors of resilience. So if we look at this pyramid, it sort of gives you the size of the problem. There are about 120 million newborns every year in this world. But we are particularly interested in these top of the pyramid babies that are these extremely preterm babies. They're about a little bit less than 1 million a year. Or also in this middle category where there are about 2 million babies every year that are already born sort of after 7 months instead of 9 months of gestation. And of course they have a higher mortality, so there's clear loss of human capital. But what is even more important is that many of these children will have moderate to severe long-term disability. And that, of course, even if it's a milder form and it's just with learning disabilities, it is as a big cost to both the child, the family, and the society. So clearly these early life events, and they can also start intrauterine in the womb of the mother, because when a fetus has a stressful environment in the mother's womb, the baby will not grow normally. We call this intrauterine growth restriction, and that actually also has similar effects as a preterm birth on brain development. So 1 in 2 of these extremely preterm infants will develop a developmental delay that is significant for his later life. So really we would like to know, how do these early life events affect brain structure and function? Just to illustrate a little bit what they go through are these images. You see they are equipped with technical devices. They often have tubes into their stomach, but they also have devices that go into their trachea, or then just nasal CPAP, which makes a lot of noise, mechanical noise of the water bubbling. And so these babies are exposed to a lot of stimuli in a sort of completely random way, unrelated to their personal feelings. So when we think about that, and we think about the basic roles of brain development, we know that there is an early sort of post-conceptional part of the early layer formation of the brain, so the neurogenesis and the neuronal migration, which is generally finished around the 4th to 5th month of gestation. This is, of course, a highly genetically controlled process, and it's usually non-sensory driven and experience independent. But then in the blue phase, the pre-wiring phase, where we get to establish all the networks and synapses and actually make certain neurons survive and others die physiologically, we start to be sensory sensitive, and there are a coexistence of transient laminar structures in the brain that are very much linked to this early activity, for example, in the subplate. When we then go into this green phase, and you see it starts around the 6th, 7th month of gestation, which is exactly around where our preterm babies are born, we become into the phase of the activity-dependent plasticity. So where the survival of synaptic contacts are highly dependent on the endogenous or even the environmentally driven experience and activity. This goes parallel, of course, with the important cortical folding that is taking place around 6 to 7 months. When these preterm babies are born, our brain is still completely smooth and there are no cortical gyri or sulci. So with this together, you can see that in this phase where the transition of the experience independent to the more sensory, sensitive driven and experience dependent plasticity is where the environment dramatically changes. It's also recently been shown that this activity drives myelination itself, generally a genetically controlled mechanisms of development through the oligodendrocyte, but it's clearly only going to start to be very prominent if there is activity in the network. So with that, we explored now for many centuries the role of magnetic resonance techniques to study non-invasively the brain development. We do this by giving the babies milk. You see it here in the stomach, so they don't get any drugs. They just sleep after feeding. And of course, there are devices that help them to keep temperature and safely studied. We can study myelination. You see this nicely here in this kind of preterm baby. You see about this amount of myelin in the images. When they get to term and they start to have nice flexion extension, you see the sensory motor tracts to be myelinated. And when they start to be able to make functional, correct, controlled movements, they have a full myelination of the sensory motor tracts. You can see also here that the full-term baby or the preterm baby with some injury has a very different pattern of myelination, for example, here in the posterior limb of the internal capsule. So if we go back to this pre-wiring phase, what is behind this pre-wiring in a more specific way? Well, it's the establishment of the brain connectivity that starts very early on with the thalamocortical fibers and the external capsule, but also the uncinate fasciculus is an early fascicle to mature. Then come the long cortical and the cingulum, and at the end, the short corticals or the U fibers. And again, prematurity occurs in a time where these connectivities are established. And when you look at these brains, clearly the connectivity in these brains are different than to the ones in the full term, so grown inside the womb. These are evaluations at school age where you see in the red colors all connectivities that are weakened by preterm birth, and the blue ones actually are improved. And you see that the weakened ones touch specifically the corticobasal ganglia thalamocortical loop, but also some short corticals, especially in the prefrontal cortex. When you then relate these weakened connectivity to actual cognitive performance, you see that many of them actually significantly relate to specific functions either in the attention domain, in the social resolution domain, or even generally in the cognitive evaluations. When we summarize them in more specific detail, we see two networks that are particularly touched, the dorsolateral prefrontal and the lateral orbitofrontal cortex. Please go to a poster session on Wednesday by Lorena Freitas, who explores this orbitofrontal cortex function, or the limbic and the paralimbic cortex with the amygdala. And that, of course, relates to many neuropsychiatric disorders that these preterm infants in blue are at much higher risk. You see the prevalences are much higher, including autism. So when we want to know what's the background of these disorders, we need to know more about the early development of the cognitive functional networks. And the ones that we're interested in are, of course, the proto-default mode network or the central executive network or even the salience network. They are increasingly present throughout development. And we recently looked at the difference between full-term and preterm at term equivalent age, and we could find with a connectivity matrix approach that really one central network that is very much decreased by preterm birth is the salience network, together with these co-active networks like the orbitofrontal cortex and the auditory network. So clearly there's a lot that makes that these brains are not connected the same way. And given that the prefrontal and the limbic system is impeded the most, we can think that maybe it's related to these environmental factors that are so stress-prone in the preterm brain. So how can we induce resilience to these early life events and stressors? One way would be to try to modulate this activity-dependent plasticity with a meaningful intrinsic or extrinsic multisensory activity. And that's what we try to do by using music. Music is known to actually activate many areas in the cognitive domain, but also in the socio-emotional domain, where there are areas of predilection to find actually weakened connectivity in the preterm population. So clearly music might be a tool to use in this sensitive period. And so we designed these little hats with the little earphones stitched into them and looked at how these babies would react to little pieces of music. Now, of course, they would be given a music that is adapted to them, and you hear it now a little bit in the background. It's a very simple tune of harp and punji and actually some bells, and they were given to the babies in a behaviorally meaningful way, in that they were given one type of music to fall asleep, one to wake up, and one to be interactive. And they were exposed to that from 33 weeks onwards to term equivalent age, and then we compared them to a group that had these same earphones but didn't listen to the music, and the full term group. The first study we wanted to know is, do they actually process this music? Is this just an auditory stimuli and goes nowhere else in the brain, or do they process the music? We used for that the psychophysiological interaction analysis to estimate the context-specific changes in coupling between the seed region, which is the auditory cortex, and the rest of the brain. And what you can see here is the specific activations that the preterm music group had compared to the preterm control group. And you see that there are areas where we compared the original heard music towards a 40% faster played tempo-modified music. So clearly areas that are known to be involved in tempo processing are activated, but also areas that are like the stratum or the middle cingulate that are known to be activated when a music or something is familiar or more arousing and pleasant. And this was only true for the preterm music group and not for the preterm non-music or the full-term group. So clearly they seem to have learned their tune. Then we looked at the structural development, looking at the seed-based tractography approach, looking at specific tracts, and also at the volume of amygdala. And these you see in red, the preterm no music, green, the preterm music, and blue, the full term. FA clearly moves in the direction towards the full term group in the overall assessment of the different regions that are outlined here. And the mean diffusivity goes the other maturing direction. Then we looked at tracts that are particularly interesting in the context of socio-emotional processing, for example, the uncinate fasciculus. And this also clearly moves towards a more full-term group for the FA, whereas the mean diffusivity, the preterm is already lower and the green is also lower. So that's a part that we need to see how this induced maturity would be related to the intervention. The amygdala volume, we know that amygdala volumes are reduced in preterm populations, and they seem to also move in the right direction. And then the last bit, we wanted to really look at the functional networks. So are these functional networks that we define to be reduced in the preterm population affected by such a meaningful multisensory stimuli? And we did this by using resting state with a bivariate estimator of accordance. We only used accordance for this analysis, not yet discordance. And you can talk to Jalel Mescaldi, who is here on poster session on Thursday about the technical details of his approach, and actually could show that indeed the salience to thalamus and proto-default mode network and the salience to superior frontal auditory or even sensory motor networks was actually very much improved by the music intervention. So the salience network, which is relevant for the detection of behaviorally relevant stimuli and then generates the appropriate cognitive behavioral response, reacted to this behaviorally salient intervention that we performed. And this could well have a relevance for future autism or executive dysfunction that we, of course, have to test in these children. So with that, we can say the music intervention, it restores some of the functional connectivity abnormalities. So this was taken up by National Geographic, and they were talking about an innovation that will revolutionize the future of medicine. That's, of course, an overstatement, but I think it shows how MR studies might have contributed to changing in some way the care of preterm infants, and that personalized and precision medicine is also arrived in neonatology. And with that, I thank, of course, all the bright PhD students like Lara Lordier, Joanna de Almeida, and Seraphim Lucas, and together with Jalel Mescaldi, who performed these studies. And thank you for your attention.